The toxicity test practiced in this chapter has used the embryo and larva stages of bivalves, such as like hard clam and oysters. The first metal mercury at the level of 11 microgram per liter concentration, it produced a 50% of oysters embryo develop abnormally, and oyster begin to grow retarded at the level of 4 microgram per liter. What is interesting is that the embryo responded sensitively. However, the metamorphosis larvae was not was less sensitive response. The Mosa embryo was exposed to barium acetate and strontium chloride, and the highest sensitivity was observed during the gastrula stage, and the blastula and the trochophore larvae were less sensitive. Mm, Tributyltin, TBT, which is commonly used as like anti-fouling paints and shit, it reduces the reproduction and development of the clam. The major population reduction in Northern Europe were due to use of a TBT products. The TBT primarily affect reproduction rather than embryo development. However, many bivalve species suffer reproductive and developmental failure at the same time. The Pacific oyster larvae, which is found in Arcachon Bay, which is in southern West France, the embryotoxicity and genotoxicity of the copper and cadmium were studied. Embryotoxicity was measured by calculating the percentage of abnormal D-shaped larvae, and genotoxicity was evaluated with DNA strand break using the COMET assay. After 24 hours of exposure, significant increase of the percentage of the abnormal D larvae and DNA strand break were observed from 0.1 microgram per liter for copper and 10 microgram per liter for cadmium. The result indicates a strong and positive relationship between embryotoxicity and genotoxicity with copper and cadmium, and further, it also indicates that copper can cause a abnormality and DNA damage in oyster at environmentally relevant concentration because it responded in very low doses. It can naturally happen. Mm, The effect of cadmium on embryo development of cephalots are rarely researched and found little effect of concentration up to 100 microgram cadmium per liter has effect on hatching success. However, at 1000 microgram of cadmium per liter, the eggs develop abnormally and eventually they all died. Cephalot can resist up to like 1000 microgram cadmium per liter, while oyster can only resist like 10 microgram cadmium per liter. Thus, the cephalopods can resist more than oyster. One of the first organics is a pesticide. Unfertilized egg of an adulis were exposed to the insecticides like savin, carbaryl, and hydrolytic product like 1-naphthol for an hour. The result shows that the most sensitive stage was when the first polar body appeared right after the fertilization. When the EC50, which is like median effective concentration, was 5.3 milligram per liter. The developmental effects included disjunction of the blastomere, retardation of the development, and unaligned and asynchronous cleavage. The embryotoxicity and the genotoxicity were evaluated with two pesticides, metolachlor and ilgarol. The Pacific oyster, again, larva was used. The ilgarol is supposedly less toxic substitute for TBT in anti-fouling paints. After 24 hours exposure, significant increase of the percentage of the abnormal D larvae and the DNA strand break were observed at the 0.01 microgram per liter for both irrigarol and metolachlor. A strong and positive relationship between embryo toxicity and genotoxicity was found with these two pesticides. This study indicates that irrigarol and metolachlor can induce abnormality and DNA damage in oyster at environmentally relevant concentration. They are, however, less toxic than TBT. In contrast, glyphosate-related herbicides have very low toxicity to C. gigas embryos. Even they produce a aminomethylphosphonic acid, which is lethal to embryos or D-shaped larvae. There's no effect on embryo larvae development up to like 1,000 microgram per liter of glyphosate-related herbicides. The second organic matter is oil. In 2013, Bellas and friends investigated possible changes in toxicity after weathering of petroleum. They evaluated the toxicity of the water accommodated fraction obtained from standard fuel oil in natural setting for weathering process for a period of 80 days. And they used sea urchin and mussel embryos. So they recollect how they were during the Exxon Valdez. This research also showed the increase in WAF toxicity in weathering. The median effective concentration after 80 days lowered 80 times than the day one, while the AH, aromatic hydrocarbon concentration, slightly decreased. The sea urchin growth reduced, and the differences in toxicity between fresh and weathered fuel could not be explained by total aromatic hydrocarbon content, but the differences can be explained by particular polycyclic aromatic hydrocarbon presence. Thank you.
Hello everyone. Now let's move on to section 6.3.3 CECs. The contaminants being tested were phthalate esters or PAEs and titanium dioxide on the abalone Haliotis diversicolor. PAE exposure at high concentrations leads to reduced hatchability, increased malformations, and suppressed metamorphosis of abalone larvae, as seen in figure 6.9. As we can see in the figures, the phthalate esters showed a dose-dependent reaction to the abalone embryos. Titanium nanoparticles, on the other hand, cause no development effects in exposed abalone embryos under 2 milligrams, but caused hatchability inhibition and malformations when the dose was increased to over 10 mg. It was also shown to increase the toxicity of TBT 24, likely due to TBT adsorption into the nanoparticles followed by internal aggregation. Next, let's talk about hypoxia. Some gastropods and bivalves incubate their embryos in their pileal cavity for around 4 weeks, after which they emerge as larvae or juveniles. Hypoxic conditions can cause a drop in the pH of the intrapileal fluid, which can impact shell formation. Hypoxia can also affect deposited egg masses and can slow development, increase mortality, and reduce size at hatching. But if photosynthetic organisms associate with the other organisms in hypoxic conditions, like the sea snail, the negative effects may be reduced. if they receive enough light. An increase in temperature made squids and cuttlefish increase their oxygen consumption 28-fold for the squid and 11-fold for the cuttlefish. Elevated catabolic activity and faster oxygen depletion within the egg capsules also intensified metabolic suppression in late embryos. Increasing the temperature only reduced the time to hatch in a snail species, but when a temperature change was paired with hypoxia, it slowed development, stopped hatching, and cell secretion, likely due to diversion of carbonates for internal pH balance. Lastly, we have climate change and ocean acidification. Experiments have shown that acidification affects many marine invertebrates and impairs fertilization, cleavage, larval development, settlement, and reproductive stages of marine calcifers, including echinoderms, bivalves, corals, and crustaceans, as seen in Figure 6.10. What Figure 6.10 shows us are the negative physiological changes that occur as the partial pressure of CO2 is increased between the samples, and that the larval stages of sea urchins and bivalves were the most affected. Figure 6.11 shows us the life cycle of benthic calcifers, and as we can see, most echinoderms and mollusks begin their shell synthesis during their larval stage, explaining why they were the most affected by acidification. Ocean warming can also affect mollusk embryos. Newly spawned squid egg masses were subjected to temperatures just 2 degrees Celsius higher than their normal temperature, which caused premature hatching and metabolic suppression. Even after hatching, they displayed a metabolic burst correlating with temperature. Heat shock proteins HSP70 and HSC70 and antioxidant enzyme were seen in hatchlings, which reduced the negative effects of heat compared to embryos. Lastly, warmer conditions can increase oxygen consumption as well, increasing metabolism and requiring more food for energy. That's it for our presentation. Thank you.